Hello and welcome students. Today we are going to discuss how to cure and salt meat. First, we will discuss about curing process. Curing refers to application of nitrite or nitrate with salt and other ingredients to meat for improved preservation. Curing is not used as much today for preservation of meats, except in very salty items like country ham and prosciutto. Rather, it provides for the development of a characteristic flavor and color and also provides protection against Clostridium botulinum outgrowth. Although curing was used in ancient times as a means of meat preservation, it's employed now more for flavor and color development. The classic meat cure ingredients are sodium chloride, nitrite, nitrate, and sugar, sucrose or glucose, with sodium chloride being the major ingredient. In addition to these, some products may contain curing adjuncts such as phosphates, sodium ascorbate, potassium sorbate, monosodium glutamate, hydrolyzed vegetable proteins, lactates, or spices that results in the distinctive characteristics of cured meat. First, we will come to meat curing ingredients. First, we have salt. Salt is the most basic ingredient in all curing brines and dry mixes. Without the inclusion of salt, the curing process would be impossible. Originally, it served as a preservative by dehydration and osmotic pressure, which inhibits bacterial growth. Salt still functions as a preservative in the country-style cured meat product. The main function of the salt in other cured products is to add flavors and is essential in solubilizing muscle proteins. Granulated or grain salt was commonly called corn, thus term corned beef. Due to its undesirable harsh flavor, salt is often used in conjunction with various sweeteners to provide a mild flavor. Additionally, salt improves yields and influences textural characteristics. Food-grade salt, that is of the highest purity, should be used in meat curing practices. The amount of salt used in brines and dry mixtures can vary considerably. Usually the level is self-limiting. Extreme levels of salt make the products too salty, and too little salt can result in inadequate protein extraction. Most curing brines range from 35° to 85° salometer. Brine strength is checked by use of salometer, a hydrometer that is graduated to show the extent of saturation of brines. Now we will come to the sweeteners. There are several types of sugars that may be used in formulating curing brines and dry mixtures. Some examples of commonly used sugars are sucrose, dextrose, and corn syrup. These constitute the most commonly used sweeteners by the meat industry today. Sweeteners function to counteract the harshness of salt and provide roundness and enrichment of flavor. Sucrose also functions as a preservative, but the levels required to provide this effect would probably render most cured meat too sweet. Also, sugar provides a surface color characteristics of aged ham. If caramelized sugar is used, both brown and white sugars can be used. The amount of sugar used is self-limiting due to its sweetening power. Now the third is nitrate or nitrite. Though attempts to preserve and extend the available meat supply, early humans used salt as a means of preservation. Nitrate impurities in salt used for this purpose led to the observation of reddish-pink colors in the meat. This observation led to the direct addition of nitrate to preserve meat in order to maintain a uniform color. During the past century, research demonstrated that nitrate added to meats is reduced to nitrite upon degradation by bacteria. Hence, it was decided that nitrite was responsible for the fixation of cured meat color through its reaction with the water-soluble proteins, myoglobin and hemoglobin. The properties nitrite contributes to the cured meat are indeed unique. The original function of nitrite in the meat curing was the production of the cured meat color. In addition to color, nitrite works as an antibacterial agent. It retards the progression of oxidative rancidity and it has a profound influence on the flavor of cured meat. The use of nitrates in food preservation is controversial. This is due to the potential for the formation of nitrosamines when nitrates are present in high concentration and the product is cooked at high temperature. The usage of either compound is therefore regulated. For example, in the United States, the concentration of nitrates and nitrites is generally limited to 200 ppm or lower. They are considered irreplaceable in the preservation of botulinum poisoning from consumption of cured dry sausage by preventing spore germination. After curing ingredients, we have curing adjuncts. In addition to the commonly accepted curing agents, the meat processor in the curing process commonly includes several other adjuncts. Examples: ascorbates and erythrobates, phosphates, starch and hydrocolloids. Problems over the decades associated with color and yields have been the primary motivation for the inclusion of these ingredients. First, let's take ascorbates and erythrobates. Ascorbic acid C6H806, termed vitamin C, a water-soluble vitamin, is often used as a curing acylator. That is, it helps speed the conversion of nitrite to nitric oxide to hasten the development of cured color in rapidly processed cured meats. 1 part of ascorbic acid is equivalent to 1 part of erythrobic acid. The benefits of using ascorbic acid in the cured processed meats are: A. Curing time can be substantially reduced. B. A more uniform color will result throughout the product. And C. Better color and flavor can be maintained during storage, distribution and display. Additionally, salt, low acidity, levels, high temperature and freezing accelerated color fixation. Another possible benefit of using ascorbate is the blocking effect of the compound on the formation of N-nitroso compounds. The USDA for curing purposes approves the following forms of ascorbates: ascorbic acid, sodium ascorbate, erythrobic acid and sodium erythrobate. Sodium erythrobate is the most widely used form of ascorbate due to its low cost. Federal regulation permit the addition of 550 ppm of ascorbic acid or erythrobic acid or the equivalent molar level of sodium ascorbate or sodium erythrobate in each 100 gallons of curing pickle. Now we come to phosphates. The use of alkaline phosphates in meat curing is widely employed by the meat industry. The various phosphates used in meat industry have been used primarily to decrease shrinkage and the degree of purge in processed meats. Phosphates appear to present a mode of action that is twofold: A. Equalating the pH of the meat and B. Solubilizing of muscle proteins. At the isoelectric point of meat, pH 5 to 5.5, fluid retention is at a minimum. Thus, an additive that significantly evaluates the meat pH should increase the fluid retention of isoelectric meat. Phosphates are essentially the salt forms of phosphoric acid. There are 2 recognizable classes of phosphates: A. The orthophosphates containing a single phosphate, anion, and B. The polyphosphate containing 2 or more phosphate anions. The following phosphates are approved for use in curing pickles: disodium phosphate, monosodium phosphate, sodium metaphosphate, sodium polyphosphate, sodium tripolyphosphate, sodium pyrophosphate, sodium acid pyrophosphate, sodium hexametaphosphate, dipotassium phosphate, monopotassium phosphate, potassium tripolyphosphate, and potassium pyrophosphate. Now we have starch and hydrocolloids. Starch is a multifunctional ingredient manifesting properties that can be applied to numerous food products. Starches contribute texture enhancement, binding properties, usually water, and improved mouthfeel to meat products. All of the characteristics are dependent on origin of the starch and its performance capabilities. The most common starch used by the meat processors are extracted either from cereals like corn, wheat, rice, or tubers like potato, konjac, and tapioca. Each type of starch has inherent characteristics that can be utilized to produce specific textural and sensory properties in cured meat products. Some hydrocolloids such as carrageenan, lotus bean gum, xanthan gum, guar gum, and pectins are also used in cured meat products because they provide enhanced textural improvements in low-fat or fat-free sausage products. Now the role of nitrite or nitrate in meat color. Nitrite or nitrate are used in the curing meat to counteract the undesirable effects of salt on color. The color of cured meat is generally dependent on three factors: A. The concentration of myoglobin in the muscle tissues. B. The degree of conversion to the nitrosyl pigment. And C. The state of muscle proteins. As indicated, both nitrite or nitrate are used in meat curing for color stabilization. The end result is the same in either case, although the pathway for stabilization of color by nitrite is more direct. Nitrite requires one less step in stabilization of color, as shown here. All of these reactions are non-reversible. Nitric oxide is the first pigment of cured meat. Upon the application of heat, it is converted into nitrosochrome, which is denatured protein and exhibits the characteristics pinkish red color of cured meat. Now there involves the bacterial inhibition. Nitrite addition to meat is responsible for the traditional and distinct color and flavor of products such as ham and frankfurters. An additional major benefit is the retardation of Clostridium botulinum growth and toxin production. Botulism was the first reported in Europe as a disease caused by consumption of sausage products. The word botulism was derived from the Latin botulus, meaning sausage. Current research efforts have suggested possible inhibitory mechanism by which nitrate inhibits C. botulism. These include A. Formation of an inhibitory substance from nitrite and other meat components. B. Nitrite or intermediates acting as an oxidant or reductant on intercellular enzymes or nucleic acids. C. Restrictions of iron or other metals essential to C. botulinum by nitrite, thereby interfering with the organism metabolism or biological repair system. And D. Reaction of nitrite with cell membrane to limit the metabolic exchange or substrate transport. Metal sequestering agents such as EDTA have been demonstrated to enhance the inhibitory activity of nitrite whereas excess iron caused a decreased inhibition. Another theory is the conversion of intercellular iron, which is essential to C botulinum, to an unavailable form after reaction with nitric oxide. Recent evidences indicates that they may also inhibit E. coli, Salmonella, and Campylobacter if insufficient quantities. Now comes the flavor. Nitrite commonly used in production of cured meats. It's a major contributor towards development of characteristics cured meat flavor. This function of nitrite in cured meats has been linked to any specific flavor component. Sodium nitrite alone is a very potent flavor enhancer in the cured meat. Salt is a catalyst for increased fat oxidation and rancidity. Claims have been made that salt is the major factor responsible for cured meat flavor, rather than nitrite or the absence of oxidative rancidity. It should be noted, however, that salt pork and bacon have quite different flavors, suggesting a positive role of nitrite in flavor formation. Now the lipid oxidation. Nitrite commonly used in the production of cured meats is a major contributor towards retardation of lipid oxidation. The effect of nitrite on retarding development of rancidity is probably due to the same reaction that is responsible for the color development. The heme compounds of mussels contain iron ions that are quite active as catalyst of lipid oxidation. When nitrate reacts to form cure pigments, iron is retained in the heme, usually in the reduced Fe2 positive form, making it unavailable as a catalyst for lipid oxidation. This reaction therefore probably accounts for the prevention of warmed over flavor in cured meats. Nitric oxide as a free radical can also terminate lipid autooxidation. Unsaturated fatty acids are targets of lipid oxidation, and the nitrosation of double bonds also could decrease lipid oxidation. The addition of 50 ppm of sodium nitrite has been shown to reduce lipid oxidation products by nearly 65%. Nitrite intake and nitrosoamines. Concerns have been expressed over the potential hazards of using nitrite in cured meat since the compound can react with amines, especially secondary amines, to form N-nitrosoamines, which may be carcinogenic. Used under the existing regulations, nitrite is not considered a health hazard. There are reported rare toxic episodes, mostly due to accidental overuse. The lethal nitrite dose is 300 mg per kg of body weight. Nitrite can react as a vasodilator and hypotensive agent. It can reduce the storage of vitamin A in the liver, and it can interfere with thyroid function. It is firmly established that nitrite can oxidize hemoglobin to methemoglobin, lowering the blood's ability to transport oxygen. This anomaly is called methemoglobinemia. It can be fatal and most commonly occurs in infants. Now we come to the methods of curing. The meat industry employs several methods of meat curing. However, they are all modifications of two basic procedures: dry salt curing and pickle curing. Dry salt curing is the oldest method. Evolution of the curing art eventually led up to the more modern method of pickle curing. First method is dry salt curing. Dry salt curing is a method dating back to prehistoric times and was the first curing method practiced by humans. The process uses salt alone or sometimes in conjunction with nitrite or nitrate. The moisture is drawn out of the meat by curing agents and drained off, leaving the meat drier and harder. The product is usually laid skin side down, and all areas are exposed, lean and plastered with the curing mix. This process is commonly used for fatty cuts such as jowls and fat backs. The second method is dry country-style curing. Curing ingredients employed by dry curing generally are salt, sugar, nitrate and nitrite. The common practice is to rub the mixture into the surface of the products and place the products on shelves in a curing room held at about 36 to 38 degrees Fahrenheit. A general rule of thumb is 11/2 to 2 days per pound of ham. Bellies up to 2 inches thick will cure in 14 days. It's also prudent to remove and overhaul or recoat the product with curing mixtures halfway through the process. Dry curing may also be used in conjunction with brine injection for some products. The product is usually pumped with about 10% of saturated brine, and the balance of curing ingredients is applied as a dry rub. Third method is brine soaking. Brine soaking is probably followed by dry curing and was often used commercially for several years. Meat pieces are placed in curing brine, and the cure is allowed to penetrate the entire portion. This process is relatively slow and spoilage may develop before the process is completed. Some items such as corned beef briskets and tongues may still be processed this way. However, the practice is becoming less frequent. Fourth method is curing pickle injection. The industry standard today is the injection of curing pickle directly into the meat pieces. Internal injection of curing ingredients enhances efficiency and promotes a more rapid and uniform distribution of the cure throughout the products. There are 3 basic methods used to accomplish pickle injection. A. Artery pumping, B. Stitch pumping, and third C. Multiple needle injection pumping. Smaller establishments may use artery and stitch pumping because these techniques are slow and labor intensive. Large establishment processing thousands of pounds products daily use multiple needle injection. Now we will read these methods briefly. First, artery pumping. This method introduces curing pickle through the arterial system of the product. Hams are the only product cured in this manner. The single needle is inserted in the femoral artery and the cure is pumped into the ham. Great care must be taken during fabrication to preserve intact the femoral artery. The process does not lend itself well to high speed, high volume production and is therefore seldom used today. B. Stitch pumping. Stitch pumping utilizes a single needle that has several openings. The operator inserts the needle in the meat piece in many different locations to deliver the appropriate amount of pickle. An experienced operator is required to evenly distribute the curing solution. After pumping, an equilibration period is often required to allow the cure to evenly diffuse throughout the product. C. Multiple needle injection curing. This method is widely used by the meat industry today due to its speed and effective distribution of curing agents throughout the tissue. It enables processors to manufacture large quantities of product daily. These injectors may be configured to pump either bone-in or boneless product of various sizes and shapes. Pumping speed and the volumes of pickle injected may be adjusted as needed. Pump head pressure must always be closely monitored. This should prevent excessive tissue disruption and formation of pickle pockets in the tissue. Now we come to the massaging, tumbling or mixing. After the curing pickle has been introduced into the meat pieces, some types of mechanical engineering is usually applied. That is massaging, tumbling, or mixing the meat pieces for various lengths of time, usually under some degree of vacuum, may carry out this process. These physical processes are employed to extract salt-soluble proteins and improve and accelerate the distribution of cure throughout the product. Now first we have massaging. Massaging involves frictional energy resulted from the meat pieces rubbing together. Meat massages are valves that contain a mechanism for the slow stirring of meat pieces. The stirring arms or paddles made are set to various configurations. The agitation time can vary depending on the product, but generally ranges between 3 and 6 hours. The massaging process is a gentler form of mechanical energy input and works well with softer textured products. This is especially important if the product is to retain a whole muscle appearance. Now the tumbling. Tumbling is more severe type of physical treatment. Tumbling involves the use of impact energy resulting from meat pieces falling and striking baffles or paddles contained in a rotating drum. Tumbling is usually carried out under vacuum to offset the potential problems of incorporating air into the protein exudates. Most products are loaded into tumblers immediately after injection with the curing pickle and tumbled from 3 to 6 hours. Longer cycles have been investigated, but due to severe limitations of time in commercial environments, this time frame works best for maximum efficiency and product quality. As with massaging, tumbling is carried out to extract proteins for binding, to enhance tenderness and juiciness, and to increase product yields. Now the third is mixing. Mixers generally have some type of paddles or ribbons that rotate around a metal shaft. They may or may not be equipped to hold vacuum, and they impart rather vigorous mechanical energy to the product. Short mixing times are usually the rule because longer mixes tend to both tear or hold muscle products and smear coarse ground products. Mixers are most often used in manufacture of sausage products, but some whole muscle products are processed in this manner. With this, I conclude today's topic of discussion.